In this class, we are going to learn about rDNA technology. rDNA technology. rDNA technology stands for Recombinant DNA technology. Recombinant DNA technology. Recombinant DNA technology. The first step is extraction and isolation of DNA. Isolation of DNA. This is the first step in rDNA technology. We have to isolate the DNA first. So we have to employ the cell wall digesting enzymes. Cell wall digesting enzymes. Cell wall digesting enzymes. Or employed or applied. The cell wall digesting enzymes are applied. And the genetic material is extracted. So then protease enzyme is applied. Protease enzyme applied. Who are digested? Proteins in the nucleus digested. Proteins will be digested. Next, RNase enzyme is applied. RNase enzyme is applied. So who are getting digested now? RNase digested. RNA digested. So now we'll get the pure form of DNA. Okay. Chilled ethanol. Chilled ethanol is added to precipitate the DNA. To precipitate the DNA. So we are going to add the chilled ethanol so finally DNA is getting precipitated. Okay. Next, second step. If we need a fragments, then obtaining DNA fragments is the second step. Obtaining DNA fragments. Obtaining DNA fragments. That is the second step in rDNA technology. Second step. So to produce the DNA fragments, what are the enzymes that we should apply? Restriction enzymes. Restriction enzymes. Restriction enzymes. Restriction enzyme. This is nothing but restriction endonucleases. Restriction endonucleases. So this restriction endonuclease, the restriction endonuclease is a specific enzyme which will make the DNA into fragments. Okay. Next, the third step is isolation of the desired DNA fragments. Desired DNA fragments. Isolation of desired DNA fragments. Isolation means separation. Separation. So how they are isolated? Based on their length. Based on their length on agarose gel medium. Okay, by the process called electrophoresis. Okay. So DNA fragments, gel electrophoresis method only we should use. DNA fragments separated. DNA fragments gets separated. So the DNA fragments are separated based on their length. Based on their length on agarose gel medium. On agarose gel medium. Agarose gel medium. The process is gel electrophoresis. Gel electrophoresis. Gel electrophoresis. And followed by this separated DNA fragments. Then how to visualize them? Right? You have to visualize them, right? So the separated DNA fragments are visualized by using ethidium bromide, by using ethidium bromide followed by exposure to UV radiations. Okay, first we have to stain them with ethidium bromide, used for staining actually. The ethidium bromide is used for staining them, then visualizing them by using UV radiations. By using UV radiations, they are going to visualize. Okay. So finally, the DNA fragments are clearly visible now. Then we can extract. Okay, these DNA fragments now extracted. Now extracted from the gel. From the gel. From the gel. That process is called elution. This process called elution. 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 Elution process. Okay. The next step is very simple. Fourth step, amplification. What is the method we use? What is in the amplification? First of all, increasing the number. Okay, multiplying the number. So amplification of desirable amplification of desirable DNA fragments. Desirable DNA fragments. Amplification of desirable DNA fragments. What is the method we use? PCR method. What is PCR method? Polymerase chain reaction method. PCR. Polymerase chain reaction. Polymerase chain reaction. Polymerase chain reaction. So here PCR method will be having 3 steps. In 3 steps, the PCR method is performed. So the step number 1, denaturation. The first step of PCR method is denaturation. So what is meant by denaturation? Denaturation is nothing but the double-stranded DNA is made into single-stranded by heating 94 degrees Celsius. So DNA fragments will become single-stranded. Okay, DNA single-stranded only, no? Right. At 94 degrees Celsius. By heating at 94 degrees Celsius, you are converting double-stranded into single-stranded. That is denaturation. Okay. So each single-stranded DNA is nothing but template strand. Each single strand, each one is nothing but a template strand. Each one is nothing but template strand. Template. Each one is called template strand. Next. Second step, annealing. Annealing. So annealing step will be involving the two sets of oligonucleotides. Oligonucleotide chains. Oligonucleotide chains. Oligonucleotide chains are added. Are added. Are added to the medium. To that particular medium, two sets of oligonucleotides are going to be added. Okay. So they will become complementary now. They will become complementary. They will become complementary. They will become complementary. And followed by the base pairs will be complementary, and thereby they will form DNA. Okay, double stranded. And this should be done at the temperature of 50 degrees Celsius. 50 degrees Celsius. First, what you are doing, the double-stranded DNA, you are making into two single-stranded DNA by heating at 94 degrees Celsius. Now what you are entering? Two oligonucleotide strands. Two oligonucleotide strands. These are two oligonucleotide strands. You are adding. That's it. Okay, at a temperature range of almost 54 degree, 50 to 54, 50 to 54 degrees Celsius. You are adding these two oligonucleotide strands to this template strands. This template strands, template 1 and template 2. Template 2. Got it? So to these, against these two template strands, you are adding oligonucleotide strands. This process is nothing but annealing, and it should be done at the temperature range of 54 degrees Celsius. And the next step is extension. The third step is extension. Extension step. Extension. This extension will be including the addition of an enzyme to this medium that is called a Taq polymerase. Taq polymerase enzyme. Extension. This Taq polymerase enzyme, actually it is why it is called Taq polymerase, I'll tell you. This T for thermus, T for thermus, AQ stands for aquaticus. AQ stands for aquaticus. So this enzyme is extracted from the thermus aquaticus, so that's why it is called Taq, and it is a polymerase enzyme. Same like DNA polymerase, it will promote the polymerization of the nucleotide. Polymerization of the nucleotides, extending from 5 prime to 5 prime of the template to 3 prime of the template. So this should be okay. This promotes, this enzyme promotes polymerization of the nucleotides, polymerization of the nucleotides from 5 prime to 3 prime. Okay. And this should be done at the temperature range of 74 degrees Celsius. 74 degrees Celsius should be done. Okay. Followed by the fifth step. So PCR, by that PCR is completed. Now fifth step, ligation of the cloning vector. Ligation of cloning vector. Ligation of cloning vector and DNA fragment. And DNA fragment. Okay. So this leads to the formation of recombinant DNA. This leads to the formation of recombinant DNA. The isolated DNA fragments, the process I am telling you now, the isolated DNA fragments, uh, and the cloning vectors and cloning vector and cloning vector like plasmids. Okay, any vector like plasmid. Okay. Mixed. Okay, both are mixed and then incubated. Okay, whatever the desirable DNA that we isolated as a DNA fragment, and it should be joined with the cloning vector only. No, otherwise we cannot transfer it. So we selected that vector, and to this isolated DNA fragment and vector we mixed and then incubated by using DNA ligase enzyme. By using DNA ligase enzyme. DNA ligase enzyme. It helps in attachment of nucleotides, right? And further, due to mixing and incubation of these DNA fragments and cloning vector, finally recombinant DNA is going to form. Recombinant DNA going to form. Recombinant DNA forms. Okay. So finally we got recombinant DNA. Still 50% of the process we have completed. Okay. Next step is sixth step, the insertion of rDNA into host cells. The insertion of rDNA into host cells. Insertion of rDNA into host cells. This rDNA that we got, that is nothing but recombinant DNA only, no? rDNA, okay? rDNA here I am writing. Finally, we got an rDNA. Okay, 50% of the process completed. Now, the insertion of rDNA into host cell is the most important step. Here, we use different methods of transferring this rDNA into host cells. First method is vector mediated vector mediated technique, vector mediated transfer or technique, whatever. Vector mediated transfer. I write transfer here. Vector mediated transfer. This vector mediated transfer means if the host cell is a bacteria, if the host cell is bacteria, condition I'm writing. If the host is bacteria, suppose you want to transfer rDNA into a bacteria. That's why I'm telling you. The host cell is bacteria. The host cell is a bacteria. Then the rDNA of the um, bacterial cells are incubated with the okay, then rDNA of the host cells incubated with incubated with 50 mm 50 mm divalent CaCl2 solution, calcium chloride solution, CaCl2 solution, CaCl2 solution, at a temperature range of 42 degrees Celsius. At a temperature range of 42 degrees Celsius. It will increases the formation of transient pores. It leads to the development of a pores. Transient pores formed. Transient pores are formed in bacterial cell wall. In bacterial cell wall. In the bacterial cell wall. Transient pores are formed in the bacterial cell wall, so that the rDNA enters into bacterial cells. That's it. So that the rDNA so that rDNA enters bacteria. So that rDNA enters into bacteria. So this is one method. Next, second method. The one more method, non-vector mediated transfer. Non-vector mediated transfer. That means in the first method, by using vector, how to transfer rDNA into host, we studied. Now, without using the vectors, we can transfer rDNA strings. So, we will study those techniques. Non-vector mediated transfers. Okay. First one, micro injection. Micro injection. Micro injection. So this micro injection is nothing but the syringes are used here to deliver the rDNA directly into nucleus. Okay. So micro injections, which includes the syringes, micro syringes, directly delivers rDNA. Directly can deliver rDNA. Can deliver rDNA into the nucleus into the nucleus of host cells. Nucleus of the host cells. So this is one method. Okay. So usually done in the animal cells. It is usually done in animal cells. Usually done in animal cells. It is usually done in animal cells. Micro injection method, mainly animal cells it is done. Okay. And coming to the second method after that, electrophoration. Electrophoration method. Electrophoration means protoplast of the protoplast and rDNA. The protoplast and rDNA placed in protoplast and rDNA placed in ionic solution. Placed in ionic solution. Electro, I told, right? Electro. So the protoplast of host cells and rDNA both are placed in an ionic solution. Ionic solution. Exposed to higher low voltage. Exposed to higher low voltage. High or low voltage. Higher low voltage. So what it leads to? It leads to the formation of again transient pores on the plasma membrane. Okay, the transient pores will be developed on the plasma membrane. Transient pores develop in plasma membrane. Transient pores develop in the plasma membrane. Transient pores develop in plasma. So further, the rDNA will enter into the cytoplasm. Then the rDNA, then the rDNA enters into cytoplasm of host cell. So this is one more method. The rDNA enters into cytoplasm of the host cell. This is one more method. Next, third method is also there. Biolistics method. Biolistics. It is also called gene gun. Gene gun. Biolistics are also called gene guns. Okay, micro projectile guns. Micro projectile guns. Okay. Insertion of rDNA by coating. It includes the insertion of rDNA by coating by the insertion of rDNA by coating this coating with 1 to 2 micrometers of gold or tungsten particles. Coating with 1 to 2 micrometers of gold or tungsten particles. Gold or tungsten particles. The insertion of rDNA by coating with 1 or 2 micrometers of the gold or tungsten particles, and bombarded with bombarded with the high velocity helium high velocity helium, so that we can target the cell to target the cell. Okay. The insertion of rDNA by coating with 1 or 2 micrometers of the gold or tungsten particles, and then it is collided with, we allow this to collide with the high velocity helium particles so that it can target the cell. Okay? Target the cell. Okay. Generally, it is done in the plant cells. Usually it is done in plant cells. Next. So by this, non-vector mediated methods completed. The first one, micro injection. By using syringes, you can directly deliver the rDNA into nucleus of the host cells. And second method is electrophoresis. What is electrophoresis? The protoplast and rDNA are placed in ionic solution and exposed to high or low voltage. Thereby, it leads to the formation of pores on the plasma membrane so that rDNA enters into the cell cytoplasm. And third method is this biolistics or gene gun method. The rDNA coated with 1 or 2 micrometers of gold or tungsten particles and allow them to collide with the high velocity helium particles to target the cell so that rDNA enters into the cell. So these all are non-vector mediated techniques. Now, culturing of host cells. Culturing of host cells. This is the most important method. After selection of the selection after selection of host cells with desirable gene After selecting the host cells with the desirable gene, the recombinant cells, the recombinant cells are cultured. The recombinant cells are cultured on suitable medium, on suitable medium, on suitable medium, on the suitable medium. So this is culturing of host cells. By using suitable mediums only, we can culture these recombinant cells. Right? Recombinant cells means the host cells with desirable DNA. Okay, recombinant cells are cultured on by using suitable medium. Next step is extraction. Extraction of obtained extraction and obtaining whatever extraction and obtaining. Okay. Here we can use the specific word that we can use and extraction and then obtaining the desirable product. Desirable product. The recombinant product will be produced, right? Either it may be a recombinant protein or a recombinant product. Okay. So that is nothing but, see here, by sending the desirable DNA, you are already sent a desirable gene into the host, right? That means the host gene is nothing but trans gene only. No? The desirable gene has transformed the host gene. So that the host gene is now trans gene, right? Trans gene. The host gene is modified into trans gene. So this trans gene produces the desirable proteins. Desirable proteins. The trans gene produces desirable proteins. And these desirable proteins um, are produced or synthesized under controlled conditions. Under controlled conditions, then the gene product is extracted from the medium. Then extracted. Then it will be extracted. Okay. And followed by downstream processing that we already know. Okay, what is meant by downstream processing? We all have an idea. Okay, downstream processing. But finally, you extracted the recombinant proteins or recombinant product. Okay, desirable proteins or products, whatever it may be, you are extracting finally. Okay, products or proteins to extract it. Now, downstream process. This is nothing but a separation. The downstream processing is nothing but a separation and purification. This is nothing but a separation and purification. Separation and purification of, the biotechnological product. The biotechnological product. The biotechnological product. So this downstream processing will be including the separation and purification of this biotechnological product. Biotechnological product. So this RDNA products are formulated with suitable preservatives. Formulated with suitable preservatives to protect their consistency for a long time. So we use suitable preservatives. Preservatives. We use suitable preservatives. Okay. Next. And such products will be undergoing even sent for clinical trials also. Clinical trials. To test their efficiency and mechanism of functioning, they have to be undergone for clinical trials. It will, whether if it is a drug or medicine, definitely it has to be undergone clinical trial. Okay. Directly we cannot sell in the market. Okay. Suppose you take coronavirus vaccine, it is under clinical trials only. Human trials are going on. Animal trials already completed. Now human trials are going. After human trials, if it is safe to consume this particular vaccine, then it is being approved. It will be approved, and then it will be sent to the market. Okay. So this is all about the RDNA technology. Okay. Thank you.